Today's topic for discussion will be on Bio Boost, which will be taken in two parts. The first part will cover the basis on photobiomodulation therapy. Hello, everyone. I am Dr. Divya Bharati Selvam from Department of Prosthodontics, SRM Dental College, Ramapuram. So the first, we need to know why this topic is required for discussion. So we all know in order to enhance with the osseointegration related to implant therapy, both the laser and LED, why it does stand out? Because the conventional surface modifications, yes, I do agree it is an effective one, but only pre-surgical, but it doesn't influence with the healing phase. Whereas PRF, it is an invasive biological modulation and more time sensitive, which will be varied based on the handling. Whereas photobiomodulation therapy, let it be laser or an LED, it is a non-invasive light therapy uses either low-level laser or LED in order to promote the osteoblastic activity, enhances with the ATP production, and full healing window of implant can be aided and successfully regenerated with the help of photobiomodulation therapy. And that is why we are in the need of this topic to be discussed further. So these are the references. So in this session, we'll go covering what is a PBM, history, how it works, what is the mechanism, what are the different types, how it reacts with the tissues, and what are the key parameters we need to consider, and where are we today with the PBM as a therapy overall. So first we'll get to know what is photobiomodulation therapy. It is a non-invasive therapeutic approaches which utilize a low-level light therapy or a laser irradiation in order to simulate with the cellular activity, enhances with the tissue healing, reduce with the inflammation and alleviate the pain. It operates by delivering at specific wavelength of light. Please do remember the wavelength will be generally ranged between a red and a near infrared spectrum around 600 to 1100 nanometers. That is why it is called as low level laser therapy or a photobiomodulation therapy or a light emitting diode therapy. So it will not be in a non-ionizing form, and the 8th generation will not be there when we are using with the less amount of the wavelength, and as well as the key parameters what we use for PBM, it is going to be differed from a conventional laser protocol and trigger with the biological response at molecular, cellular as well as with the tissue levels. So it is previously known as cold laser, low level laser, low power laser. As I said, 600 to 1100 nanometer without causing any thermal or ionizing effects. These are the two things you need to consider when we are giving with the PBM therapy apart from the wavelength. So what are the key parameters? So we got to know wavelength 600 to 1100 nanometer, and ideally for a photobiomodulation therapy, the power output will be very, very minimal. That is 50 to 500 milliwatts. The total energy density need to be delivered to the target tissue. For the simulation activity, it should be 2 to 8 joules per centimeter square. And effect type, it is going to be non-ionizing and non-thermal, and its primary interaction with the tissues are efficient to be cellular stimulation and with the tissue healing. So, photobiomodulation therapy history. Previously, in the year of 18th century, Niels Rayberg-Fensen is a pioneer who used with the red and the blue light. In 1903, Dr. He has been awarded with the Nobel Prize because of utilizing a chemical rays which has been absorbed from the sunlight could have an bactericidal effect as well as simulating tissues. So these light therapies or photobiomodulation, it is not something new. So it's been for the years over there where even from the sunlight we can have an interaction with the tissue and it has an bactericidal effect. For this, he has been awarded. So, pioneer of the laser therapy is Dr. Henry Master. He is the first person to indicate with the uses of low-level laser therapy, where he did a study on the rats in by taking out the hair and whether the regenerative capacity is there for the PBM therapy by utilizing the laser or the LED wavelength, the minimum level. He is the first person. So, with that, in the year of 1960s, we developed with the laser therapy. 70s to 80s, its expansion came forward to the medical and the dental research. In 1990s, we termed and we developed with the new terminologies, and there is a recognition started for this therapies. Around 2007, its standardization and applications got expanded, and now we are integrating into the mainstream dental as well as the medical fraternities. So how this therapy works? So photo means light, bio means a living cell, a modulation means there will be a change in the tissue response. When the tissues are in a very much stressed state, more amount of an oxidative stress will be produced by the cells and less amount of ATP production. Because of that, the cell got repaired. Whereas when you're going to give the PBM therapy to the cells, what it is going to do, it is going to enhance with the ATP production and reduce with an oxidative response, and thereby its response is going to be better in nature and there will be an healing happens, right? So how does the mechanism of PBM is going to work? So as I said, this is something not a new concept where with the light energy will be able to produce with the energy or with an ATP or with the growth enhancement. So every day humans, animals and plants, we utilize with the energy which has been absorbed from the sunlight for normal function and growth. One of the few example is your photosynthesis process which we observe in day-to-day basis. In the same way, there are certain biological photoacceptor molecules going to be present in our body, such as rhodopsin, which is present in eye vision pigment. That is why when the laser treatment we are giving, it is better to wear with the goggles in order to prevent its interaction with the rhodopsin pigment. Hemoglobin, myoglobin, and cytochrome C oxidase. So the key difference, the 660 nanometer, it is going to target the direct mitochondrial, whereas the near infrared, it will interact with the cellular membranes, leading to a deeper and biological effects. So laser unit with an increased synthesis of ATP, DNA and RNA soluble protein mediators, it will enhance with the cell proliferation during the wound healing period, as well as it will enhance with the phagocytosis process. Whereas in terms of a systemic effect, it is initiated in a distant target by circulating to the soluble protein mediators, and this is how the cells will get absorbed with the help of the photons released from the PBM unit. So what are the primary things we can expect, such as direct events like reactive oxygen species release and biomolecular conferential changes can happen. Indirect events like modulation of antioxidant, ATP production, and no dissociation, and nitric oxide dissociation and cellular repair. The secondary phase, which includes with platelet aggregation, mast cell activation, cytokine release, and growth factor, and which suggested that it has an anti-inflammatory effect also. Clinically, it is going to impact in the wound healing and neovascularization, angiogenesis, and reepithelialization. So thereby the PBM, it can be successfully utilized in the tissue repair and especially with the regenerative capacity into the dentistry field. So what are the different types of photobiomodulation therapies are there? So based on the source of the light, we can categorize them into an low level laser, light emitting diode, super luminous diode, and broadband light luminous. Between these four, the first two, they are more commonly used both in the medical as well as in the dental fields. There are previous studies suggesting that after crossing beyond certain millimeters of tissues, the light source, it doesn't have any changes in its interaction. So it is all to be primarily played with the wavelength rather than the type of the light source. So these light sources are preferred because of their specific wavelength, coherence property, and control energy delivery. So as you could see with the image differences, the coherent laser light, it is going to be coherent, whereas LED, it is going to be incoherent. Laser, it is going to be a pinpointed delivery. LED, it is going to be of a broader applications. So all these changes in the properties between the laser and LED, its effect going to get varied rather than the light source as such. So this is what the previous studies has been suggested. So cell response is primarily depend on wavelength, mechanism, and dose, not with the source of the light. Please do remember with this thing. So what are the key parameters we need to consider for an effective tissue interaction? So the optimal window, you all know, 600 to 1100 nanomillimeters with a deep purification with minimal absorption by water and maximum interaction be required on the cytochrome C oxidase. Whereas the soft tissue surgery, it is going to be just a vice versa condition where we require more of a water interaction for an effective changes in the soft tissue hard with the hard tissue. So correct wavelength, it is in the first law of photobiology. So without a proper absorption, we can't expect with any kind of a reaction. If energy density or the fluence, it's going to be adequate, you will have an adequate reaction. Too much, it is going to produce with it. Too low, you'll not able to see any results. So optimal parameters is sufficient enough. So Azar Zam suggested with few of the laser interactions, which one to be an ideal? Less than 500 milliwatts per centimeter square, a total energy density to be 2 to 8, either in a continuous or in a pulsed mode. The target, it's going to be in cytochrome C oxidase are surrounding with the cell membranes, and biological response time, it is going to be longer in PBM therapy when compared to the other treatment because the dosage is going to be very minimal. So this dose-dependent effect of PBM, it was proposed by Hanan School's curve, where there is an insufficient energy, no response, weaker or a lower stimulus, you can expect a biostimulation. When the dosage is going to be really high, you will see an inhibitory effect, a worsened effect. So be very specific what is the ideal parameter for the tissue you are working on and for which effect you are working for it. Right. So where are we today with this photobiomodulation therapy? So it is known to have a direct and indirect effect. Indirect effect like a neuroendocrine immune system and with the circulatory system. Now we came to the concept called Pacer principle. In laser, we use the photons to amplify the light sources through the stimulated emission principle. Pacer, where you use stimulated emission to accelerate electrons instead of the light source. So this is where we are today, where we are gone to an upward principle called Pacer. So with that, I complete the first session. Thank you. We'll continue with the session 2, where it will be talking on the implant integration with the PBM therapy.